Welcome back to a new video of Sunanda's tutorial. In this video we will discuss about intermediate filaments. So it is a type of cytoskeleton. So apart from microtubules, microfilaments, we have intermediate filaments as our cytoskeleton. Okay, so intermediate filaments have a diameter of about 10 nanometer, which is intermediate between diameters of two other principal elements of cytoskeleton. Actin filaments, that means microfilament, consist of actin filaments, and that actin filaments is of about 7 nanometer diameter, and microtubules have 25 nanometer diameter, but intermediate filaments have 10 nanometers. So it is present intermediate. So it is intermediate between microfilament and microtubules. So this is called as my intermediate filaments. In contrast to actin filaments and microtubules, intermediate filaments are not directly involved in any kind of cell movements or motility. Instead, they appear to play basically a structural role by providing mechanical strength to cell and tissues. So it provides structural strength, mechanical strength to the plant. It does not help in the motility or movement. Next, the proteins which are involved in intermediate filaments. Actin filaments and microtubules are polymers of a single type of proteins. Microfilament consists of actin protein and microtubule consists of tubulin proteins. But intermediate filaments consist of a variety of proteins that are expressed in different types of cells. So more than 50 different types of intermediate filament proteins have been identified and classified into 6 major groups based on the similarities between their amino acid sequences. So we have type 1 to type 6 types of intermediate filaments. Type 1 and type 2 consist of two groups of keratins. Type 1 is called hard keratin. Next, some are soft keratins. The hard keratins are used to production of hair, nails, horns in case of animals. Then soft keratins are abundant in cytoplasm of epithelial cells, with different keratins being expressed in different types of cells. Next, type 3 intermediate filament proteins include pimentin. They found in different types of cells including fibroblast, smooth muscle cells and white blood cells. Type 3 protein called desmin, which is expressed in muscle cells. Type 4 type of intermediate filament protein include neurofilament proteins. They are particularly abundant in axons. Okay. Next, type 4 protein is expressed in earlier stage of neuron development or nerve cell development. Then type 6 is called nestin, is expressed even earlier during the development of neurons in the stem cell of central nervous system. Next, type 5 type of intermediate filament protein are nuclear lamins, which are found in most of the eukaryotic cells, because nuclear lamins in the nuclear membrane is present in the eukaryotic cell, because prokaryotic cell lack the nuclear membrane. Okay, so you have to remember that which kind of protein or which type of intermediate filament protein act on which cell. Okay, next, so these are the list of intermediate filament proteins and their site of expression, their size. Next, despite of considerable diversity in size and amino acid sequences, Various intermediate filament proteins share a common structural organization. Of all of the intermediate filament proteins have a central alpha helical rod domain of approximately 310 amino acids. This central rod domain is flanked by amino sequence and carboxy terminal domain. So this is the basic structure of intermediate filament protein with N terminus and C terminus or carboxy terminus. One is amino terminus, one is carboxyl terminus. Which vary among different intermediate filament protein size, sequence and secondary structure. And this is the central rod or alpha helical rod. It plays central role in filament assembly, while variable head and tail domain presumably determine the specific function of different intermediate filament proteins. So this is the basic structure of intermediate filament proteins. Okay, next, assembly. How the intermediate filaments get assembled. The first stage of assembly is the formation of dimer, in which the central rod domain of two polypeptide chains are wound around each other in a coil structure similar to formed by myosin 2 heavy chains. Then so at first two chain like this form a dimer. Two monomer associate with each other to form dimer. Next, dimer then associate in a staggered antiparallel fashion to form tetramer. When four monomer associate with each other or two dimer associate with each other, they form tetramer. Next, because they are assembled from antiparallel tetramer, both ends of intermediate filaments are equivalent. Consequently, in contrast to actin filaments and microtubules, intermediate filaments are apolar. They do not have plus and minus end, but microfilament and microtubules have plus and minus end, but these lack plus end and minus end. So this is the diagrammatic representation of assembly of intermediate filaments. At first, one monomer or polypeptide chain associate with other to form a dimer, then tetramer formed in an antiparallel pattern. In one dimer, amino end is here and carboxyl end is here, but in another dimer, carboxyl end attached to the amino acid end and amino acid end towards the carboxyl end of the other. So in this way they attach with each other and finally form one protofilament. The central rod domains of two polypeptides wind around each other in coil structure to form dimer. Dimer then associate in a staggered antiparallel fashion to form tetramer. Tetramer associate end to end to form protofilament and laterally to form filaments. Each filament contains approximately 6 protofilaments wound around each other in a rope-like structure. So this is one filament consists of 8 protofilaments like this. Okay, so this is the assembly of intermediate filaments. Next, intermediate filaments are generally more stable than actin filaments or microtubules and do not exhibit dynamic behavior. They do not frequently change their shape. They do not frequently assemble and disassemble they do not show dynamic behavior associated with these other elements of the cytoskeleton. However, intermediate filament proteins are frequently modified by phosphorylation, which can regulate their assembly as well as disassembly within the cell. One of the example is the phosphorylation of nuclear lamins, which results in the disassembly and breakdown of nuclear envelope during mitosis. So during mitosis, karyokinesis occur where the breakdown of nucleus into two for two daughter cells. So at that place, this disassembly of this intermediate filament occur. So cytoplasmic intermediate filaments such as vimentin are also phosphorylated at mitosis, which can lead to their disassembly and reorganization in the dividing cell. So in the mother cell at first they get disintegrated, and in daughter cell they again get assembled. So more precisely we can say that this intermediate filament can get disassembled by the process of phosphorylation. Okay, next the function. So at first, cell adhesion. At the plasma membrane, some keratins or desmin interact with desmosomes and hemidesmosomes via adapter proteins. Next, associated proteins. Filaggrin binds to keratin fibers in epidermal cell. Plectin links to vimentin to other vimentin fibers as well as to microfilament to microtubule, and myosin and kinesin is being researched and is suggested to connect vimentin to tubulin via motor protein. So these are generally associated proteins, helps to associate one protein with other. Keratin filament in epithelial cells linked to desmosomes through placoglobin, desmoplakin and desmoglein and desmocolins. Desmin filaments are connected in a similar way in heart muscle cell. So these are generally associated proteins. So it is all about your intermediate filaments. Okay, I hope you really enjoyed this lecture video and will get benefit from this. If you liked it, please hit the like button and please share with your other friends, and please subscribe to my channel to get more important videos like this. Thank you.